Okay, so this is lecture 11 of Cog Neuro midterm 2. And this is the fourth one in midterm 2, but 11 overall in the class. Okay, and so the first thing to talk about is someone named Carl Lashley. And so Carl Lashley surgically separated the motor regions of mice. And so their motor regions are cut in half, and then he realizes that this doesn't affect maze learning in rats. And the only thing that does affect maze learning is the destruction of the cortex. And so if you destroy the cortex, then maze learning goes bad. But if you just split the motor region, then it won't go bad. But you have to destroy a large part of the cortex. Okay, and so the way you can remember this is you think of a man named Carl lashing at a brain, and it destroys the cortex because the cortex is what's on the outside. And so as he's lashing, he's destroying that cortex. And this is called graceful degradation or mass action. And graceful degradation and mass action is the principle that this memory is widely distributed. And so now we think that the memory is kind of all around the brain. It's not just in one part. And you can remember this because graceful degradation, then you go back to Carl Lashley, who's lashing at the brain. And then the graceful degradation, you can think of Carl Lashley gracefully whipping of the brain. And so he's just kind of sitting there, weaving his little whip, just gracefully whipping this brain. Okay, and so then you can also remember that it's mass action because a big mass of the cortex is this huge, or a big mass of the brain is this huge cortex. And so graceful degradation, mass action are the principles that memory is spread out wide over the brain, and that that was done by Carl Lashley. Carl Lashley, mass action, graceful degradation. And destruction of the cortex destroys maze learning. Okay, moving on to the next concept. The next concept is Donald Hebb, and he came up with the idea that memories are stored in cell distributions, and so basically this is LTP. And so basically what he was saying is that cells that fire together wire together, and LTP is when you need less of a stimulation to make that cell fire. And so Hebbian learning is LTP. Hebb, LTP, Donald Hebb, LTP. You can think of the president, Donald Trump, and how he can't do LTP because he's stupid. Okay, so Donald, LTP. LTP is basically learning. Donald Trump clearly never learned. Donald Hebb, cell distribution, LTP. Okay, and another thing you could think of is Hebbian learning. Think of the head and the glee people, and then you hold the L on it because Heb sounds like head, and then L for like the losers on Glee, and then it's LTP that he came up with. And so Hebbian learning is LTP. And think about that because on the heads of the Glee people, there's an L that symbolizes loser. Okay, and so then this LTP concept was proved by Lomo and Bliss in the rabbit hippocampus. And you can remember Lomo and Bliss because Lomo sounds like low mo, and the rabbit is down low in a ditch holding an L on its head. And L could be for Lomo or L could be for LTP. And then you think about just for this new concept of LTP, you can think about a rabbit in a ditch because it was rabbit hippocampus holding an L for LTP, but also for Lomo and Bliss. And low in the ditch. So Lomo and Bliss, LTP, Lomo and Bliss, LTP, Lomo and Bliss, LTP, Lomo and Bliss, LTP. Okay, so Lomo and Bliss did the LTP thing, which is basically a cellular model for learning. And so there are two parts of LTP. The first part is specificity. And so basically this is the concept that things that are not active are not going to be potentiated in this LTP. And so only neurons that are firing are going to be potentiated. And so you can think of specificity because specifically problems come to inactive people. And so specifically problems come to inactive people. And so inactive people have issues. And specificity means that inactive neurons will have issues firing. Okay, and so specificity is basically if you're not firing with the group, you're not going to be wired together. And then the second one is associativity, and that is that other active neurons are also potentiated. And so this is just saying like if neuron A fires, it's going to be potentiated with neuron B. Okay, and so once again, the two concepts of LTP are specificity and associativity. Okay. Also going along with this is LTD, which is long-term depression, and that happens after long periods of inactivity and long periods of low-frequency inactivation. So LTD, long-term depression, is going to happen if you have a long period and you don't get stimulated very much, and then it's going to be depressed. Because LTP happens when you have a bunch of stimulation and you learn. Okay, and so how do you represent this in something other than a cut of a rabbit hippocampus? Like how do you represent this in an animal? And the way you do that is in a mouse, and so this is a in vivo model, and you can remember that in vivo means in animal because vivo sounds like vivere or vivere. It's some Spanish word that means to live, or vida la vida, which is live the life. And so in vivo means that it's in like a living life form. And so a potential in vivo model for LTP is these mice that have glutamate receptors knocked out. And so glutamate, so basically if you want to represent LTP in vivo, you use a glutamate or NMDA receptor knockout mouse. And the way that they tested this was in a spatial memory task. And so another thing to remember is that spatial memory is in the hippocampus, because how do you find your way around campus? Well, you find your way around campus with the hippocampus. Okay, and so just to go over it again, potential in vivo models are the glutamate NMDA receptor, and potential LTP models are the glutamate NMDA receptors, which is in vivo, and then remember that the hippocampus has to do with spatial memory, which is why they tested these knockout glutamate NMDA mice receptors. And so, like, the receptor was knocked out, so they couldn't receive glutamate or NMDA. Like, they could still make it, but they can't receive it. And the reason that they tested it in this hippocampus is because the hippocampus is for spatial orientation. And so what actually happens in LTP? Well, there's some morphological changes. And so one of those could be that the synapses would grow in size. And so there's 4 options. So once again, grow in size. So that's the first one, grow in size. And growing in size means that the vesicle is going to bulge up. Like, think of it as like getting fat, because growing in size doesn't just mean that it's bigger. Like, you have to think about what that means, and that means that there's going to be more vesicles, and so there's going to be more little neurotransmitters to be transmitted, and also there's going to be more postsynaptic receptors to receive these more vesicles. And so grow in size, there's just like more ammunition being fired out, more things receiving these bullets. And then the second thing you can do is develop clefts, and the third thing is to split. And these kind of look the same. I'm not super sure what the difference is between clefts and splits, but just think about it. Cleft lips look kind of split, and so it's cleft and split, because it's not really cleft and it's not really split. It's both of them. So cleft and split, and that's in addition to the first one, which is grow in size, and the fourth one is grow from scratch. And so going back and reviewing, the morphological consequences of LTP are going to be that you grow in size, so more vesicles, more receptors, and that you could develop clefts, or you could split. And cleft and split they seem like the same thing. I'm not really sure what the difference is. Or also, you could grow from scratch. And so that's just like a completely new dendritic spine. Again, that's what those are, dendritic spines. And then just to go over what we already looked at, Carl Lashley discovered LTP, not Carl Lashley discovered LTP. Do not think that. Carl Lashley discovered that the memory representations were kind of all over the brain. You can think about this because it's like a man whipping the outside of the brain all over, and he did this by separating the motor regions, but that didn't work. And the only way to get rid of memory is to destroy a huge part of the cortex. And so that's graceful degradation and mass action. Donald Hebb discovered LTP, and this was proved by Lomo and Bliss in a rabbit hippocampus. And the 2 crucial aspects of LTP are, 1, specificity, which means that inactive neurons are not potentiated, and associativity, which is just the reverse. And so other active neurons are going to be potentiated in that. So cells that fire together, wire together. And also, there's long-term depression, which happens when there's long periods of low-frequency stimulation, and the potential models are glutamate or NMDA receptor knockouts. And so you can't receive these little glue particles, you can't receive these little NMDA things, so no glutamate or NMDA receptors in these knockout mice. And so you can think of it as like there's no mitten, like in baseball where there's the bat, the ball, and the mitten, there's no mitten to receive these glutamates. And the glutamates are kind of like the little baseballs. And they tested this by performance in spatial memory tasks. And finally, in this section, you need to remember that the hippocampus has to do with spatial memory, because how are you going to find your way around campus? You're going to use your hippocampus. So hippocampus, spatial memory, hippocampus. Okay, and then finally, the morphological changes in LTP are 1, grow in size, which means more receptors, more vesicles. 2, develop clefts or split. And 4, you can grow from scratch.